Anyway, so you're like, well, why is this guy giving me a lecture on immunology 1 and 2 for the next 45, 50 minutes? The reasoning is that I've done most of my research in translational immunology, whether it be with malaria, brucella, Burkholderia pseudomallei, other pathogens in the past, including Cryptococcus. So I've done a lot of cool stuff, bench and translational wise. So this is probably why this lecture was assigned to me. So, but I want this to be interactive, okay? You know, if you just want to hear a lecture, you could do this on YouTube, right? But and there's nothing wrong with that. But I want to get to know the new fellows. I've already gotten to know one, and so we're at 1 and I guess a quarter. And so I want to get to know you all, and I want you all to, you know, interact with me. I'm going to ask you questions. I'm not going to sit back and just lecture to you. So the objectives are to understand the relationship between antimicrobials really in the human immune system. Okay, we're going to go over basic immunology and the principles for both innate, which is what I call basic immunology 1, and then basic immunology 2 is just adaptive immunology, just an overview, because I could sit here and probably lecture to you for over a year on adaptive immunology. So, and we don't have the temporal course for that, nor the patience. So, but again, I want to make sure that you all understand that they're not in silos. The innate immune system and the adaptive immune system are not in silos. They are connected, and they are constantly in connection with each other for not just pathogens, but also just to kind of talk to each other and see what's going on. This is also important in the realm of things like cancer, allergy, autoimmunity, and all kinds of other cool stuff. Okay, shall I begin? Okay, so I am employed for the most part at the VA. And so you can kind of think of the immune system as a hierarchy of, you know, different cell types with different actions. The same thing happens with the Department of Defense, right? So you have the Secretary of Defense who, you know, answers to the President of the United States, and there's different people, right? So there's the Secretary of the Navy, of the Air Force. You have somebody who's in charge of you know, the Army. There's an Inspector General who watches over everybody, and there's all these little you know, lines that go down from each one of them, right? And so, but I think that's kind of a mundane way to look at it. I like to look at it more like The Sopranos, okay, which is one of my favorite TV shows of all time. Even though I'm mainly Italian, it doesn't, you know, it does resonate to me with other ways. The interesting thing, if you've never seen The Sopranos, is that, so this is the main character, Anthony Soprano Sr., and he is the head of a mafioso family in northern New Jersey. And so he's got two lives, right? So he's got the life on the outside where he's dealing with business, and those are the guys on the left, right? And then you have his home life, which includes his uncle down at the bottom, his wife and children, and also, funny enough, his psychiatrist. And so he's got to balance these two things. And there are threats coming in from whether it's, you know, the authorities, whether it's other crime families, other things. And so, you know, Tony Soprano has to balance these things. And that's basically what the immune system does, is it balances, you know, the threat of an invasion with also not making sure that there's collateral damage within the family, which is the body, right? And so this is kind of the way I look at it. And there will be Sopranos references as we go through this. I put this slide up not to scare the living crap out of you, but in reality, for you to understand the complexity of the immune system. This is in a patient who has malaria, Plasmodium falciparum malaria to be exact. And you could probably see my cursor, that this is a dynamic process, right? And so whether it's innate things like, you know, nitrous oxide that's being, you know, emitted by red blood cells that are infected, and you know, go through the endothelium here, and then there's pro-inflammatory cytokines that are released from macrophages in the spleen that are harboring red blood cells that are infected. This generates cytokines that start both local inflammation and B cell presentation to Th1 cells and Th2 cells, right? And so this causes, again, this cascade of these types of cells starting a cytotoxic T cell response to cells that are infected. And then you have on this side, you know, more of a part of the immune system that goes through how homeostasis is returned, as you see not only T regulatory cells that help stop the effector response, and that's through IL-10, TGF-beta, other things. But then you have other cells, right? You have NKTs, you have natural killer cells, and then you also have gamma delta T cells, which are a very, very, very important part of the immune system. And we're just in its infancy in terms of understanding this. Questions? Okay, so this is the gross anatomy. Okay, so you've got, all of the areas in the body which have mucosal surfaces, right? Whether that be small intestine, that could be respiratory, and whatnot. And so this is connected to the lymphatic channels. And then from there, this is also connected to the spleen, connected to lymph nodes themselves, and also the thymus. Bone marrow is also involved, obviously, because that's where B cells come from and a lot of different innate cells. So, but this just shows you that we're going from the macro level down to the micro level. So for basics, right? Simple forms of life have basic forms of immunity. So, they'll have some innate, cells to stop infection and inflammation. But again, as invertebrates and vertebrates evolve, you'll see that the immune system increase in complexity because there's different areas of their bodies that they have to protect. Not, different from, let's say, a helminth, which is basically a tube within a tube. So pathogen induction occurs through either PAMPS or DAMPS. We'll go through that. And then there's innate versus adaptive, where innate cells, their job is to stop infection locally, but also to tell the other cells locally what's going on. That includes adaptive immunity, which is adaptive immunity is a way for is a higher level of immune cell, which in this case are lymphocytes, that stop infection at different points, whether that be through antibody production, whether that be through regulation of the immune cells, or whether it be destruction of cells through apoptosis or through granzyme and perforin destruction. So again, what does innate encompass? We're talking about cells, we're talking about complement, innate cytokines, and antimicrobial peptides. Adaptive immunity is really effector versus memory. And that's really for a later lecture. I really just go through the basics here, but it is important. But please stop me before you, if you all have any questions. So innate, what does that mean? It means native or to be born with something, right? So it's the initial form of immunity before the adaptive immunity begins to prime. So in babies, this is very important, right? So in neonates who can't really mount an antibody response. And so a lot of the antibodies that are in use in babies come from milk, right? And mainly mother's milk. And so it's very important that for mothers' breasts who are breastfeeding to to have had all their vaccinations and whatnot, so that then they can impart that immunity to their babies. There's also anatomical and physiologic barriers, right? So you've got tight junctions and other things that are border things like the epithelial layer in the GI tract. There's also cilia and other things that occur in the respiratory tract to literally filter out and brush out pathogens and debris, right? So then you have molecular defenses and cellular defenses. So this could be the molecular defenses we're talking about, antimicrobial peptides, really, and then of course complement. And the cellular defenses really are the macrophages and neutrophils and the like basophils, who are on the precipice of, or the forefront rather, of infection and trying to stop it. So the presentation of antigen is also important by some of these cells, and this also activates the adaptive immune system. So there's also the regulation of the initial immune response, right? So if a macrophage can control this, depending on the cytokines that they release, right? And they also act as, let's say, and again, another Sopranos reference, the cleanup crew after a pathogen immune system battle occurs locally. So I just kind of talked about this, right? So, you've got the anatomical, physiological barriers, the gross anatomy. Also, I forgot lysozyme is also very important. the hydrochloric acid within the stomach. Innate immunity we already kind of talked about, and yes, even eosinophils are part of that. And then you have complement. You have mannose-binding lectins, things like dectin-1, which we'll go into. And then you have the adaptive immunity, which is cellular and humoral. So let's talk about the complement cascade. So it's really it's an ancient form of innate immunity, actually. And so you'll see this in invertebrates, lower vertebrates, and the like. So in humans, there are small proteins that are produced by hepatocytes, right? And so they're usually precursors. The reason they're precursors is that so they're not activated all the time, right? So this way, they're activated by certain chemokines and cytokines through a process of proteolysis, and then they subsequently start to activate throughout the body to bind bacteria. It could be yeast. It could help antibody for optimization and the like. There's different parts of this, and I'll show you a cartoon of this shortly, where you'll see the classical pathway, a lectin mannose pathway, which is really through identification of certain carbohydrates. Alternative pathway, which is enacted by certain other chemokines. And again, this could be for virus-infected cells, fungi, bacteria, protozoa. And the other part of what they do, again, is to help with optimization. This is the classic picture. This is from Nature Reviews of, and you probably remember this from med school, and you're like, good grief, here we go. I tell the fellows that everything you learned in step 1 comes back to bite you in ID if you didn't remember it well. And this is a good example of that. So the classical pathway, you've got the, you have antigen antibody, complexes binding, which is mainly through complement number 1, which is also called C1, binds to C4 and C2, goes through a convertase for C3. This stimulates an immune response to certain lymphocytes, and then also later on can convert C5 to C5A and B, which then creates the MAC complex, which is what destroys gram negatives. And again, if you have a deficiency within the complement complex, you could have problems with certain types of infections. This will be covered in a later lecture, but it's to clue, and also for things like, for bone marrow transplants, solid organ, cluing in on what problem you have with the immune system. That's either by an iatrogenic, whether it's a drug, or whether it's something that's going on, organically. You can find out what types of infections you can expect in these patients. The lectin pathway, like I said before, it has to do with certain types of carbohydrates that are identified, like dectin-1 and whatnot, and that goes from there. Alternative pathway, again, is with different other chemokines like factor B, factor D, which can start the C3 conversion through certain other mechanisms. So what are antimicrobial peptides? So they're found in various higher organisms, both invertebrates and vertebrates, okay? So a good example of this is honey. I'm gonna ask all of you, have you ever seen honey spoil? No. It's really difficult. I've only seen it once in my lifetime. And it's not through experimentation, because I was just sloppy in the kitchen, and several years ago, in fact. But it's what it does is that these peptides, they target mainly bacteria, but they can also target fungi, viruses, parasites, and even cancer cells, funny enough. And what they do is that it imparts a halogenation of that on that protein, and this causes a cationic anionic reaction on the cell wall, and this causes lysis of the cell. So basically, you have a pH reaction, an acid-base reaction, occurring at the level of the cell wall and membrane of that organism, which is pretty cool, right? So these things are in your bloodstream right now and are being pumped out by the liver. And this is why, another reason why, when people have hepatic failure, whether that's from cirrhosis or something else, or drug-induced, you will start to see certain infections pop up. And you're like, well, why? That doesn't make sense. Whether that's sequestration of iron is inhibited and whatnot. But the main reason is because of antimicrobial peptide concentration going down in the bloodstream, right? So, and again, this also stimulates certain parts of the innate immune response as well. This is a cartoon of it. So basically, you see here, there's direct killing of microbes because of not only electric static interaction, basically causing an acid-base reaction locally, but it also enacts the host immunity. And certain things can be, they can bind TLR4, TLR2, which neutralizes certain bacterial processes and certain fungal processes, and even down to if there's double-stranded and single-stranded DNA, which has to do with autoinflammation, but also when you start to see these kinds of things hanging around for RNA, the body starts thinking that there's viral particles around, and so then it starts to troll for viruses, which is important in probably not only viruses that affect whether it's COVID, it could be other things that this could also impart some type of immune response as well. This is from a Cell article. So then there's PAMPS, right? So what are PAMPS, right? So it's not somebody who can't say the word PAMPS. It's pathogen-associated molecular pattern molecules. And what they are is, and I'm going to read this straight up, they're a diverse set of microbial molecules that share a number of different general patterns or structures that alert immune cells to destroying, introducing pathogens. So these are actually parts of a pathogen, whether that's a bacterium, fungus, it could be certain signals on the outside of helminths that tell the immune system there's an invading organism there. Okay, and so again, this is well established. There's pattern recognition receptors, and where do we do this? We see this mainly through TLR, right? So TLR2, TLR4. This is what binds LPS and the like, and helps tell the immune system what's going on. Excuse me. So for DAMPs, they're a little different, right? So damage-associated molecular patterns. This is the reverse. So this is when a host cell knows that it's infected or it has turned to the dark side, basically, where it has become cancerous. And so these proteins are, excuse me, are on the surface of the cell, are recognized by the immune system because of these proteins, and then subsequently an immune response is initiated. Okay, so what examples do we see of this? You're not going to recognize some of these things, but HMGB1, some nucleotides, but ATP is definitely one, right? So if a cell is actively trying to fight off an infection within its corpus, and the cell lyses, there's going to be things that are released from the cytosol into the local environment. If there's ATP around, which means there's energy left over, that means that this is a doomsday signal to the immune system to stop what's going on. When do we see this? Again, not only in things like cancer and viral infections or intracellular pathogen infections, but also even things like autoimmunity and neurodegenerative diseases like MS. This is a good example of the PAMPS, right? So you see the pathogen, these little things are released, whether it's LPS or whatnot. These things are recognized by certain immune cells, like in this case, an antigen presenting cell, which then gives it to a T cell and tells the adaptive immune system what's going on. So again, the DAMPs are cells that are damaged, picked up by local cells, dendritic cells, macrophages, neutrophils, and then and these things then alert the T cells what's going on. So the way the way that this from this journal, from the International Journal of Nephrology, kind of puts it, there's strangers and there's dangers, right? So PAMPs are telling you what's going on with strangers, and then DAMPs tell you what's going on with your own people and that there's dangers. So let's go into phagocytosis and what is it, right? So it's a basic process by which a cell senses, ingests through a process of endocytosis, internalizes that other cell, parts of the cell, or even particulate matter. So then there's different types of phagocytes, right? So you've got professional phagocytes, right? So again, this goes through the whole Sopranos reference, right? You have a guy that does this all the time. You have a guy that you hire, right? So the professional phagocytes, things like neutrophils. Yes, even eosinophils can phagocyte stuff. monocytes and their lineage. So free macrophages, dendritic cells, resident macrophages such as Kupffer cells or splenic cells, osteoclasts and microglia. They're all resident macrophages. And then you have non-professional phagocytes, right? So fibroblasts can also do this, epithelial cells. And then interestingly, yes, lymphocytes can do this too. B cells can do this. And of course, innate natural killer T cells, especially gamma delta T cells, can not only present antigen, we'll see this later, they can also destroy cells like a CD8 cell, but they can also phagocyte bacteria and fungi to clear the area locally. Are there any questions at this point? Okay. So main focus is, again, is to clear the pathogen locally. Secondary focus is to opsonize some of this, some parts of this, whether that means it's been retained by antibody and complement, or opsin, again, the purpose of bringing this into a cell, let's say in a splenic macrophage, and figuring out what's really going on. And again, what is this? These are PAMPS. And so the tertiary focus is to present antigen to the T helper cells, which is through whether it's MHC2, which is an HLA, and you can tell those different T cells what's going on locally and also systemically. There's also a quaternary focus, which is to clear the area of damaged apoptotic cells. Again, if those cells are signaling through DAMPs. So neutrophils, right? So they're granulocyte lineage. They're stimulated by IL-8, which is now termed CXCL8. They're phagocytic pathogens. They stimulate lymphocytic T cell responses as well. And they also release nets. What are nets? And no, I'm not talking about, something that the fishermen use. What are nets? Not all at once. Mm-hmm. Come on, ladies and gentlemen. We have a lot to cover. Good grief.
I don't know what it stands for, but it's something like when the neutrophils are dying and like whatever's inside the neutrophil like comes out and like tries to diffuse like the pathogens, and that's kind of what forms the pus and purulence, something like that. Like the gut outside.
Outstanding, Dr. Sadiq. So yes, it's exactly what it is. So there's different, but they don't have to die necessarily to do it, right? They could be a slow death. It doesn't. They don't have to. This it's not a blaze of glory explosion, right? So what they'll notice is that if the neutrophils are outnumbered basically, and this occurs in a pus pocket, right? So this occurs when there's a lot of staph, where, and that's why you have abscess formation. You'll have the cell, this neutrophil, it'll dissolve its nucleus, all right? And all of the chromatin, all of its DNA, and certain its granules, which contain perforin and granzyme, all interconnect with this in the histones. Then the cell kind of starts to throw these things out, and it starts to just capture stuff, right? It's almost like a Venus flytrap, and it just catches these organisms and prevents them from doing more damage locally. Now, if a neutrophil is really dying quickly, it will do this automatically and just pump this stuff out. And then from there, it will capture the organisms and create the nets. But then sometimes you'll see other phagocytes come in, pick up what's going on, and start to do the same thing. And this is part of the development of why with things like staph and other pathogens, you have an abscess formation. Does that make sense? Other questions? This is a very important concept. Okay, silence. Excellent. Okay, so eosinophils. So they're also granulocyte lineage. They're stimulated by IL-5, and you're like, okay, why is he? Okay, why? Well, this is all important for a lot of different reasons. You can measure some of these cytokines, but you can also see how it stimulates the other parts of adaptive T cell lineage. Okay, so they release cytotoxic granules toward protozoa and helminths. All right. They can phagocyte the remnants of these pathogens to see what's going on, whether or not the protozoa or the worms are healthy. If they're not, what's going on with them. And again, this can also has a feedback loop with lymphocytic T cells. So let's go to macrophages. So macrophages are part of the monocyte lineage. Okay, they're amoebae that don't divide. Isn't that an interesting thing? So if you think about it, amoebae, free-living amoebae, they divide. Okay, but they do the same things that macrophages do. Okay, so they reside in different parts of the body. It could be liver, spleen, CNS, lung, dermis, GI tract. With the GI tract, there's lamina propria, Peyer's patches, and they also serve in bone. Right? They can be a Trojan horse for certain carriers of pathogens. Can you give me examples of this?
Mycobacteria.
Mycobacteria. Excellent. I see where your heart is, Dr. Sadiq. What else?
Malaria.
Listeria.
Yeah.
What?
Listeria.
Listeria. Okay, excellent. That is what else?
Malaria, like Plasmodium.
Okay, that it could. That's rare, but yes.
Chlamydia, Neisseria.
Okay. Less likely, but yes.
Legionella, right?
Legionella. That's perfect. That's what I was really looking for. Brucella can also do this as well. And so this is really important, right? So you can actually, let's say if you get an entamoebal infection, that can actually harbor some of these other organisms that we discussed. And so you can actually have a secondary infection. Okay. And it can also, this also occurs even with macrophages themselves, where some of these bacteria actually, hang out in macrophages and can become quiescent or make the macrophages quiescent. And then subsequently, what they can do is what? They can then cause an infection, a relapsing infection. This is notoriously important for things like mycobacteria. Listeria, less likely, but it can occur. But also with things like leishmania and Legionella and the like. Okay? Phagocytosis also occurs along with opsonization, antigen presentation. Okay. So they're also potent producers of cytokines, both inflammatory, and this goes through macrophage 1 versus macrophage 2, right? And they also engage in apoptosis. Questions?
Regarding the Trojan horse thing, what is it about those particular pathogens that you named off that is there a particular molecule or system or adaptation that they have that allows them to do that versus other kinds of bacteria or pathogens?
Fascinating question. And you're one of the new fellows that I haven't met. So, right. So, but you're asking a fascinating question. I could lecture on this for hours. Essentially, a lot of them do it through a cyclic AMP mechanism. Okay, certain organisms have genes that will use this, such as Leptospira can do it, Mycobacteria can do it, Legionella can do it, but there's also other different proteins. So Brucella classically has this one gene called FC gamma R1, and what that does is actually not only stop lysosome formation, but also causes a reverse endocytosis and retrograde through the Golgi into the rough ER, where it can actually hide in the rough ER and utilize the ribosomes within the rough ER to make its own proteins, not unlike a virus, which is highly ingenious and makes Brucella a fascinating pathogen. And so a lot of other organisms can do this too. But usually it's through a cyclic AMP mechanism that occurs through different genes to stop the lysosome from activating. Does that answer your question?
Yes, thank you.
You're welcome. So again, this is kind of what I was talking about, right? So what do these guys do, right? So we're talking about they really superoxide chemicals. They can engage in antigen presentation. They have they opsonize. They actually can help with wound healing, funny enough, through different types of growth factors through fibroblasts. Of course, they phagocytose, and of course, they can do bone resorption. That's classically osteoclastic activity, right? So on the left, you see a nice cartoon, and then you see a drawing of what it looks like, from the organelle level. You can see there's numerous phagolysosomes, a very large rough ER and Golgi body, and a large nucleus. And that's because it needs a lot of protein to do what it needs to do to patrol and also engage in T cell and B cell antigen presentation. Dendritic cells are, of course, of monocyte lineage as well. They're a little similar, where they can also do antigen presentation, but they do that much more than they do a lot of the cleanup that macrophages do, right? So they're strategic for antigen presentation for both Th1 cells and cytotoxic T cells, as well as B cells. And they do this through both MHC1 and MHC2 presentation. Do you guys remember the difference between the two? Hello? What is the difference between the two, ladies and gentlemen? Come on. This is basic biology, and you guys have got to know this. So MHC1 are on their right. So these are molecules that are on every single one of your cells, all of them. And what they do is that they are able to present antigen to cytotoxic T cells and also helper T cells. Okay, when that cell is dying or there's a problem with it, whether it's a metaplastic, dysplastic cell, neoplastic cell, or if they're infected with a virus. MHC2, and that occurs through a certain mechanism. MHC2 only occurs in antigen-presenting cells to T cells. Okay, and that occurs in dendritic cells, macrophages, and B cells. And that is when there's something going on locally. They sample it, they attach it to what's called an invariant chain, and this goes up and is part of the MHC2 complex. This can only be recognized by Th cells. This is a very critical thing, right? And so, because this is how your body tells the innate immunity tells the adaptive immunity what's going on, is really through MHC2 presentation. Okay? So dendritic cells are ubiquitous to the body, right? It's like macrophages. But where antigen detection is key, like the GI tract, respiratory tract, GU tract, they're in higher numbers. They go into this pre-stimulatory phase and then become effectors, or they become regulators, depending on their on how they differentiate. This is all the stuff they do, and you can kind of see what I was talking about, right? So, for type 1 interferons, which stimulate B cells, they can stimulate other T cells, other macrophages. Okay. There's also other things like IL-4, which stimulates Th2. We'll get into that. IL-12 stimulates Th1, interferon gamma, and also other things, right? So then you also have things like IL-15, IL-18, which stimulate NK cells and innate T cells, which can help destroy virus-infected cells or neoplastic cells. These are so non-professional phagocytes. What are we talking about here? Epithelial cells, endothelial cells, fibroblasts, and yes, microfold cells, the M cells classically in the gut. So they sample particles locally through endocytosis, kind of like, the kid in the candy store. Ooh, I'll take a sample. And they just kind of see what's going on and then tell everybody else what's really happening. So this determination is made by the cell to display antigen to whether it's NK cells, macrophages, innate B cells, innate T cells, and dendritic cells. And again, this is all done through MHC1. Okay. Now there's natural killer cells. These are a type of lymphocytes, okay, but they're a little bit different from T and B cells, okay. And what NK cells do is that they're part of the innate immunity, but they're also part of the adaptive immunity. They can respond extremely quickly to all kinds of pathological issues, right? So they're kind of like your special ops guys, right? These are the guys that, once an infection or a pathogen is identified, they go into the area locally to try and really inflict damage on infected cells and also cancer, right? So, and they bind through MHC1, okay? And then what they do is that they can also regulate T cell responses so that things don't get out of control, right? Same kind of thing like I kind of said here, right? So you have healthy cells, they become stressed, whether it's because they're bound by antibody, because they have antigen on them, or they blow up, or there's something wrong with them. NK cells can identify these two. They'll either destroy them, or they'll present the antigen to a dendritic cell and then cause the T cell response to kind of start. So how do the antibiotics help the immune system? So I kind of told this to the good Dr. Green 2 days ago in the ASP. So it's a numbers game, right? Do antibiotics really cure you of your infection, ladies and gentlemen?
No.
No, they don't. Okay, what they do is that they achieve appropriate kill of the pathogen. They reduce numbers locally, okay? So that the immune system can do its job. So that the immune system isn't overwhelmed, okay? But again, it's also a double-edged sword, right? So there's adverse effects of antibiotics, and there's also pathogen resistance. So the whole point is to return to normalcy. In this case, homeostasis is a scientific word, and that's what they're really trying to do. Does that make sense? So again, antibiotics don't cure you of infection. It's your immune system. That's why, because if you go to Moffitt, you see people who have like 2 neutrophils running around, and you give them antibiotics, that doesn't necessarily cure them of the infection. And so again, it demonstrates how important and critical all the aspects of the immune system are, right? So this cartoon kind of illustrates that, right? So what I equate antibiotics to is bioterrorism. It's bioterrorism for bacteria, fungi, protists, and helminths. And what they do is that they cause a problem with the organism. So it's they either reduce the numbers by outwardly killing them or causing them to be paralyzed in a way. And then subsequently, the immune system has an easier time regulating on the infection. So we're going to go into part 2, okay, which is more the adaptive immune system, right? So any questions right now? As I have a sip of coffee. No questions. Oh, nothing? Okay. Just as a side note, Tony Sirico, who played Paulie Walnuts, he just died recently. He was an amazing actor, and you should read about him. He was, he had a very interesting life and found his way into acting, funny enough, through prison. And it shows that people can change, and but used his talents from his history to be probably one of the most memorable characters in TV cinematic history. So let's go into basic immunology 2. Okay, so this is going to be a little shorter, only because number one, I don't want to disturb Dr. Montero's time, but also I could talk about this for hours, and you're going to see aspects of this throughout the rest of the lectures over the next 6 to 12 months. Dr. Ayler can correct me, but that's kind of what the plan is. We're going to go over adaptive immunity and the basics of it, right? We're also going to go over some innate T cell immunity, and I want this to be more interactive. Okay. There's also chemokine cytokine responses we're going to kind of go over. There's different types of naive cell function, effector cell function, memory cell function, and regulatory cell function, right? So each one of these lymphocytes, their job is they're, they don't know what's going on. Then they kind of get a role, again, kind of like on The Sopranos. There's somebody who is a doer, like the effectors. There's the guys who kind of keep the money secure, like the consigliere, the memory cells. And then you have guys who regulate on the inside, right? Who kind of tell everybody else, hey, you shouldn't be doing this. Let's take it easy. And so this way, then the immune response isn't as as robust as it could or should be and not cause collateral damage. And then again, if you recognize the different immune point deficiency, you will figure out which types of infections you will see in those patients. So this is a nice cartoon illustrating what adaptive immunity really is. But again, you have humoral immunity, which is really B cells. You have extracellular, pathogens. They bind to them with antibody. They present antigen and whatnot. Cellular-mediated immunity is, dendritic cell macrophage has pathogen. It tells the T cells what's going on. The T cells lyse the cells, and that's it. But again, I go back to this malaria slide on purpose. It's so much more complicated than that. And to understand this, the immunology literature is basically rewritten every 60 to 70 days because we're finding out so many new things about immunology. One of my mentors at the La Jolla Institute has been one of the best T cell immunologists probably in the world. I would present something to him, and he would tell me, Anthony, that data is obsolete. I said, but it was just written a year ago, and he said it's already obsolete, and it demonstrates how robust the science is when it comes to immunology. So we're going to go over B lymphocytes. Okay, and again, they develop in the bone marrow, hence why they're called B lymphocytes. Okay, so naive B cells are primed by Th cells. Okay, mainly Th2, and they differentiate into memory cells. Okay, or plasma cells. Right. So memory B cells. And once a B cell has been deemed appropriate, and it is primed, basically. It only responds to one type of antigen. Is that clear? So, when you have, and this is very important, when you're like, well, Anthony, what about certain, we give vaccines to people, they just don't mount the response. It might not be the vaccine issue. It could be a Th2 to memory B cell issue. And why that B cell is not really priming, we don't know. But it kind of tells you what's really happening, right? So plasma cells, there's long-living plasma cells, which can stay alive for long periods of time, months to years, and then some which are short-living. The short-living ones are the ones that really are the ones that are during a really, really, really high inoculum infection. And when you have to knock down pathogen quickly with antibody. Now, B cells can also be antigen-presenting cells. You can have IgM or IgE on the surface of them, okay? And then the development occurs both in the marginal and germinal centers of lymph nodes and the spleen, right? But it also occurs in the mucosal surfaces, right? So you have lamina propria, not just in the GI tract, but also in the mucosal surfaces of the respiratory tract and GU tracts. Very important, because this is how IgA is produced by some of these B cells and released, and also IgD and also other regulation effects locally. So this kind of is a nice cartoon from Nature, which kind of describes this, or is this from Lancet? I think it's from Lancet, where you've got MHC class 2 occurring on a dendritic cell, tells a naive T cell what's going on. That then meets up with a naive B cell, gives it the antigen on an antibody. That B cell is then activated and primed. It can become a low affinity plasmablast, so this can be something that just hangs out locally. These are the guys who are the long-lasting plasma cells, which can make IgA and whatnot. And then you have germinal center B cells, right? So this goes to either the spleen, or this occurs within the lymph node itself. Even though it says lymph node, it can also occur in the spleen. So it goes to a germinal center where it's where the B cell and T cell, in this case, it's a follicular T helper cell and a follicular dendritic cell, and they kind of talk as certain cytokines are released during this time, which either creates a memory B cell, or it can create a high affinity plasmablast, something that needs to go into effect now, producing a lot of IgM and IgG. Some of these guys, excuse me, can also go to secondary germinal centers and let the follicular T helper cells know what's going on locally. And then from there, excuse me, can be primed for subsequent infection. So I'm terribly sorry. My coughing is, I have a little fit here. So B cells, again, continued. So we have CD19. That is the marker that's on both mature and neoplastic B cells. So what are we talking about here? We're talking about cells that are already primed. Memory B cells or plasma cells can have CD19 on them. But also the cancers of those cells, right? So certain types of lymphomas, certain types of myelomas and other diseases of primed B cells. Now CD20 is on immature B cells, so rituximab and the like, right? So rituximab is an anti-CD20 antibody and is used in certain anti-inflammatory processes. Why is that important? So for certain types of lymphomas, especially like Hodgkin's, you use CD20, right? And so again, that's because those are immature B cells that are the cancer of those cells, right? Then question? Okay.
Regarding sorry.
Go ahead.
So regarding kind of the pathway split for all this, what becomes short-living, what becomes long-living, what becomes a memory cell, what's the nidus for that? Is it random? Is it driven by the antigen? Is it driven by what distinguishes what the naive cell becomes?
Excellent question. And you must have science background because that's exactly what happens what I'm about to explain next. So it has to do with the somatic rearrangement, right? So those B cells they're in the process of priming. They start to have this thing where their DNA, okay, they start to make different types of RNA, which can then again becomes protein. In this case, it's rearranging certain proteins that are going to be on their surface in the antibody, whether it's the light chain or the heavy chain. And this will bind certain types of proteins, carbohydrates, whatever. If a cell does not bind an antigen adequately, it goes bye-bye, unless it becomes a cancerous cell. Okay, those B cells are destroyed. So the nidus really is a random process in a way through this somatic rearrangement, also known as VDJ, which is variable diversity and joining, right? So each one of these things, some of it's variable, some of it has to do with the diversity of what those antibodies could bind, and then the joining of the light and heavy chains. Again, I could go on for this for hours, right? You've got, this occurs in the bone marrow, again, for B cells, and also in the thymus. This also occurs in T cells. Again, it happens for both immunoglobulins, which are antibodies, and also T cell receptors. There's the constant regions and variable regions, which can change. Okay, and then again, this is all based on recognition of pathogenic, antigens on pathogens. Okay, which can also be self, and allergens can also occur as this as well. So to answer your question, some of it's random, but some of it is also targeted, and it really depends on if that B cell is primed the correct way or not. And it's also cytokine stimulation as well. So does that answer your question to a certain extent? I could go on about this for hours, but I also know that I've got about 10 minutes left. So, with regards to immunoglobulins, we're going to go into just minor detail, right? So there's 5 types of immunoglobulins: IgG, which is a monomer; IgD and IgE, which are also monomers; and then you have IgA, which is a dimer, and IgM, which is a pentamer, right? So what does each one of them do? I have it on the next slide, but I want to see if you guys can answer this question. What does IgM do?
Like complement activation.
Okay.
It
really binds, it does 2 things. It really, because it's a pentamer, it can bind numerous antigens at once, right? A lot more than IgG. And this way then, it can see if there's enough antigen around to be able to sustain a memory response. In addition, it also is part of the antigen presentation for B cells to T cells. Okay, so there actually can be on the surface of the cell and then tell the T cells what's going on. Where is IgA located?
Mucosal surface.
Mucosal surface is excellent, right? So, and interestingly, you see that secretory component there, that chain that holds the two together, to the J chain. What that is actually cleaved by gonococcus, and why you cannot mount an immune response to the gonococcus is because of the fact that it can cleave IgA. Okay, IgD really occurs through antigen presentation. It's the most least common. IgE is actually more common than people think. It's on the surface of B cells, but also on the surface of mast cells, basophils, and the like. And it's really supposed to be used for parasitic infections, but really is really important in allergy now. So IgG is it's the workhorse in a way. It's the one that's produced by plasma cells in response to a subsequent infection or to other types for opsonization and other factors. Now, the question: do you only see IgM in the initial infection, or can you see it in subsequent infections?
See it in subsequent infections.
Yes. So I will tell you that, in med school, you were taught IgM is first and IgG is second. Nope. You can actually see IgM infections in certain types of robust relapsing infections. Malaria is a classic example. You can actually see that the more IgM that is released compared to IgG, those people have less severe malaria in a high endemic setting. And that's because of the fact that, again, it's a numbers game. IgG is a monomer. IgM is a pentamer. You can bind more antigen. It makes sense. The body is much smarter than you think it is. Don't think that that everything is so concrete in immunology. Again, these are things that we've learned over the years. I just kind of went through this, right? Again, IgA found in mucous surfaces, again, protects against pathogens there. Oops. IgD is part of the B cell receptor, but it really activates certain types of basophils and mast cells, but it's really part of the B cell receptor antigen presentation. IgE, again, parasitic words, but it's really allergy. IgG, everywhere. IgM is secreted first for the most part, but again, can also be in other stages of immunity as well. There are also subclasses of IgG. This can be important in certain types of immune responses if there's a deficiency. So again, we in ID order a bunch of tests, but that doesn't mean that we know why we're ordering them. Don't order something if you don't know why you're ordering it. It's a pet peeve of mine, but it's very important in academic medicine and also in medicine, period. IgG3 and IgG1 really are potent complement activation to the classical pathway, okay, and they help out with opsonization, okay? IgG2 is a weak complement activator, but it really is important in binding polysaccharide antigens in terms of things like, this is one of the antibodies that's really stimulated by the pneumonia vaccines, right? And so this is very important. It can undergo dimerization, but it's really rare. IgG4, no complement activation. It's kind of related to IgE. So if you see diseases where IgG4 is really elevated, you can actually see high allergic responses to things. Okay, and again, it can't, but it's really seen in parasitic infections, which is interesting in terms of why it's similar to IgE in terms of its structure. Okay, we're on the home stretch. So T lymphocytes. Question? Was there a question? No? Okay, sorry. So again, these develop in the thymus, hence the word T cells. Okay. They're found throughout the body, right? Lymph nodes, lamina propria, Peyer's patches, spleen, liver sinusoids, etc. I don't know why I have this here. It shouldn't be there. I apologize. It's a duplicate slide. But T cells, once they're differentiated, okay, their function becomes set, kind of like B cells. Okay, they also undergo, that's why I included it, because I wanted to include the VDJ. So it's the same thing. Again, you have the somatic rearrangement of the T cell receptor. Once that T cell receptor is primed, it really only looks for one type of antigen, okay? And once that antigen is set, it can become either a memory T cell, it can become an effector T cell, or it can become a regulatory T cell, right? Now, T cell receptor binding depends on MHC1 or 2, right? Whether it's a TH1, a TH cell, or whether it's a C or a TC cell. However, the T cell receptor can be either alpha beta in terms of its composition, or yes, something called gamma delta. And that's where gamma deltas get their name. They have a different type of T cell receptor based on the different peptides that are part of the protein. Okay. CD3 is a T cell marker in conjunction with the T cell receptor. It is present on all T cells. That's why when you get a T cell subset, you see CD3, CD8, CD3, CD4. It's because it is a flow. You do it through flow cytometry. You see exactly how many T cells there are, and then which ones are TH2. I'm sorry, TH versus TC, right? CD4 is T helper cells. CD8 is T cytotoxic T cells. And those things can either be in high concentration or low concentration, whether they're how activated they are. Okay? T helper cells are divided into different subgroups. We'll talk about that in a second. And what stimulates them? Innate T cells. So these are innate natural killer T cells, the mucosal activated T cells, and gamma deltas. They recognize carbohydrates and lipids as their antigen, as opposed to protein or peptides, which are alpha betas. Does that make sense? So if an antibody focuses on an entire protein length with carbohydrate maybe or lipid components, okay, a T cell with an alpha beta T cell receptor only looks at certain peptides within a protein. So it can recognize a pattern much more efficiently than an antibody. Okay. Now, for the other guys above them, the innate T cells, they look for carbohydrates on the surface. Okay. Whether it's a carbohydrate capsule, whether it's other proteins on this, whether other carbohydrates and lipids on the surface, to identify those cells and either tell the immune system what's going on or destroy them. This is the TH cytokine axis here, right? So again, you've got a dendritic cell, in this case, PAMPS or DAMPS. It starts the cycle where it binds to a naive T cell, probably in the lymph node. And there's other things that are that can polarize the cell to a certain direction. And then from there, that naive T cell gains function and can be one of these groups. Now, before, when I was a resident, we only really had TH1, TH2, and Treg. Now look at this. It's a mess. So you have TH1, right? So TH1 really activates cytotoxic T cells and other cells, macrophages, things locally, to destroy cells, to blow them up. So, and that is stimulated by IL-12. All right, and then then subsequently will release interferon gamma. It will release TNF alpha, and of course IL-2, which helps those T cells divide. Right, that stimulates T cell division. IL-4 stimulates the TH2 response, which causes antibodies to be released from B cells. Okay, IL-4, IL-5, IL-13. This is a very important thing, and when I talk about, pardon me, certain fungal tests, you'll wonder why. Well, if the titer is high, you would think that the fungal disease is better under control, right? Like a coccidioides test, but that's not true. It shows that the TH2 response is dominant, not the TH1 response, and thus you're producing antibody against a pathogen where it really doesn't help you clear the infection. You really need the TH1 response, and in that case, that's why that's occurring. So IL-4, IL-5, IL-13, these are cytokines that stimulate eosinophils and also B cells. For TH17, it's really a dynamic lineage, and it's stimulated through both TGF-beta and also IL-6. And you're like, wait a minute, that's a stimulatory and a non-stimulatory cytokine respectively. Yes, because it's trying to control things. And so again, What this does is this is very important for lung infections, things like mycobacterial infections, NTM, Mycobacterium tuberculosis, Bordetella pertussis, and the like. And so these cells stimulate not only natural killer T cell function, but also the innate T cells locally, like gamma delta T cells. Th9 is really has to do with autoimmune and also cancer regulations and certain infections, but again, stimulated by THF beta, and IL-9 is what it secretes, and NK cells and certain dendritic cells are their targets. Th22 is really kind of, we're still kind of figuring out what's going on with that. Th22 stimulates IL-22, which can stimulate certain types of macrophage function, and maybe also Th17 through IL-23. Then you have T follicular health. So that's stimulated by IL-21, IL-6, which will stimulate B cells, and that's why IL-4 is there. And also IL-21 also stimulates T follicular helper themselves to further identify B cells that can and prime them during a primary infection. Let's go back to the light green briefly, the Treg. Those are the cells that are the regulators, right? Literally. They are the T cells that regulate an effector response to the point where then there's not subsequent damage locally or outright kill the person, kill the patient. And so TGF beta and IL-2s are stimulated in these cells. But what do they secrete? IL-10. And I could go a whole course on different cytokines and when they peak, but it kind of is important, right? So if something like interferon gamma peaks in 22 hours, you'll know that you need that response to be quick. If IL-10 peaks at 60 to 72 hours, you know that it's being secreted to calm down that immune response. IL-34, IL-35 are also secreted as well. And so it shows that there's this yin and yang in between T cells to really regulate what's going on. Now, briefly, my time is over by about 2 or 3 minutes. We're talking about gamma delta T cells. They can do almost everything. This is why they're so freaking cool. Again, you know, iNKTs and mate cells, they bind to lipids and carbohydrates, but they can blow up cells. So they have like CD8s. They have the capacity to secrete granzyme and perforin. They have the ability to bind. They have their own T cell receptor, which is why it's called gamma delta. And so they can actually present antigen, but they can also regulate as well with a Treg response themselves. They also can enact the complement system through Dectin-1 and through other types of innate responses. And they can also stimulate different types of inflammation, whether it's IL-17, interferon gamma.